Hello from Utrecht in the Netherlands, where things are getting tighter in terms of lockdown. So it's my pleasure to introduce our second speaker, which is Henrike Heise from Jülich, as I already mentioned, and from the University of Düsseldorf, the China University. And Henrike is actually a nice example that did, both life and material science, because Henrike actually started as a PhD at the Technical University of Munich in 1999, working on metallo-organic sandwich compounds, and also, among other things, nitroxides, and we'll come back to that a little bit later, nitroxide radicals. And then she moved on as a postdoc, Alexander Pines. So she was a postdoc with Alexander between 2000 and 2000 at Berkeley, of course. And then after that, that's, I guess, also why I'm here today. So she was a brilliant co-op between 2002 and 2007 at the Max Planck Institute. And I'm surprised to see my colleague alpha assist by solid state NMR. And she did really wonderful work on this in a seminal paper that we could publish 2005, I think, on unleuctin fibril. And I guess that passion for fibrils unfolding and misfolding when she got a call for a professorship at the Institute for Physical Research Center in Jülich. And that's where she is professor now since 2007. And the title of her talk today is Protein misfolding and folding at highly sensitive and high sensitivity and resolution. And I give the floor to Henrike.
Yes, hello from Jülich, where things are still sort of under control, but not much longer, I guess. Yeah, and it's nice to see all of you again. And thank you very much for this nice introduction, and also for this opportunity to meet you all again and give a talk here. So here, I think, so is it okay?
You can see it.
Yeah, okay. And then here, laser pointer. Okay, so the title of my talk is Protein Misfolding at High Sensitivity and Resolution. And of course, yes, protein folding goes via different stages. So some proteins have a globular structure, but they also can unfold under unfortunate conditions. And some proteins never take a real structure, so we have also intrinsically folded proteins. And then, of course, when things go bad, they oligomerize via an intermediate state, and finally they deposit as amyloid fibrils. And today, so we are in our lab studying all kinds of these stages with solid state NMR spectroscopy. And today I won't talk much about this unfolded stuff. We have some nice publications, and it's all about what can you learn about unfolded, really unfolded systems with in the frozen state. So if you study such proteins at room temperature, then you get random coil chemical shifts, which are actually averages between all kinds of secondary structure elements, which are sampled very quickly. And if you freeze those proteins out, then every protein is frozen in its state. And you have a large distribution of states and a large distribution of chemical shifts, which leads to ugly and broad lines. This is something everybody who works at low temperatures has suffered from. But actually, if you overcome this by selective labeling, then you can look at different selected peak shapes and try to extract some information about that. And that's what we did. And we have already published that. So this was the work of my postdoc Baran Uluja-Yaski, who's at the moment at maternal leave, but soon she will come back. So we got some information about different states, and we could shift different proteins between membrane-bound states and also fibrillar states and see the distributions of that. And also we are trying to look at sidechain conformations in collaboration also with Flemming Hansen, who talked last week or 2 weeks ago, I think, here in this seminar. Okay, so the next stage of protein misfolding is the oligomeric intermediate. And of course, this is the most interesting, but also the most difficult state that a protein can adopt, because it's an intermediate, and that means it's not stable. And the main challenge before you can start to do structural stories about studies on that is that you somehow have to stabilize it. And of course, it's also not easy to get in a nice conformation. But my PhD student Anna König and our group tried to solve this problem in collaboration with some Lothar Gremer in our institute in Düsseldorf, and Nadine Rösner, who actually managed to stabilize that together with the prion protein, with another protein that has a potential to misfold. So it has been known for some while that prion protein can act as a receptor for beta oligomers. So these are some microscopic images which show that if you incubate some nerve cells, neuronal cells, with A beta oligomers, they co-localize with the prion protein. And also in the group of Kostilev together and Schlittmatter together with Kurt Silm, there were some studies about such a in vitro preparation of PRP and A beta oligomers, and they prepared a so-called hydrogel phase from some liquid phase separated prion protein where oligomers were added to. And they also saw some helix formation in the N-terminal region here, also in the unbound N-terminus of prion protein. And this interaction, yeah, is it bad or good? First of all, it can mediate some bad toxic signal. It can start some toxic signal cascades via the Fyn canals, and it also mediates in that synaptic dysfunction. But there's also this N-terminal N1 fragment up to residue 111 in prion protein, which can sequester oligomeric A beta and maybe also protect then the nerve cells from too many oligomers. So there are two sides on the coin. And we decided in collaboration with Lothar Gremer and Nadine Rösner on the interaction between prion protein and A beta oligomers, and to use two different constructs of the prion protein. First, the full-length construct starting from residue 23 to 230, which has two binding regions, which are lysine-rich and highly positively charged, which can then bind to the more negatively charged A beta. And here, from residue 95 to 110, there's also again a second lysine-rich region. And those two, on the shorter construct, 23 to 144, and both of these constructs can bind and sequester A beta oligomers. When they're incubated, they form complexes, and under the AFM is here the picture of the oligomers, and here of the complex, which is dust-like clouds. And so what we got, our precipitate is clearly not a hydrogel. It's a fine dust, and it's very heavy. It precipitates immediately and sinks to the floor. So it's a bit different phase of what has been studied by Kostilev et al. So, and if we adequately isotope label either PRP or A beta, then we are able to study these complexes from both sides. And that's what we did. So first we looked at the mobility of prion protein, and this is the full-length prion protein, and we looked at it from three sides. So you can either do inept excitation, this is what liquid state people do, and then the mobile signals of the mobile parts of the proteins are excited. And in those spectra, we see nothing. That's why I didn't put them here, because they are useless. Then you can do cross-polarization, where you excite only the rigid parts, and then you get the immobilized part of the protein, and they are displayed here at three different temperatures. And on the left side, we also did as a comparison a 90-degree direct excitation, and with that you excite the full protein, no matter whether mobile or rigid. And what we see here is, first, the temperature doesn't make such a big difference in the CP. And the difference between CP and direct excitation is also not so big. We see some additional signals from the background here in the direct excitation spectrum, and here also the Teflon, but that's the main difference. And apart from that, the signals look more or less the same. That means the full prion protein is more or less immobilized here. And we don't have any highly mobile parts here. Okay, we also did some comparison with solution state NMR of this shorter construct, 23 to 144, which doesn't have any structure. It's natively disordered, and the liquid state spectra were taken by Philipp Neudecker and his co-workers. And what we see here is, if we overlay those spectra, of the immobilized N-terminal part of prion protein and the liquid state spectrum, then we see they match perfectly. So it's the secondary structure is not changing a lot. Most of the black spots fall on top, exactly on top of the red spectrum, which is a solid state NMR spectrum. And here also in the glycine region, we don't see two peaks like the Kostylev group did, but we see really one peak, which is of course a bit inhomogeneously broadened, but we don't see too many features here. What we observe is for two residues, we see some alpha helix-like propensity of valine, of alanine here, and also of leucine. And to see that, alanine and leucine are very informative residues. If we look at the sequence, we see them only in this region from 130 to 133. And that means, and also the two leucines are in this region only. And that means we get some helical propensity, but only in that region. And so where we are here. And the glycines are not so much affected. That means we have the flexile N-terminus remains mainly unfolded, but we have here a region which has some more alpha helical propensity, which is also in agreement with what has been observed before. Okay, then if we take the full length prion protein and look at that, we see following changes, or we can compare it also to the N-terminal part. The N-terminal part is here displayed again in red, the borderline of that. And if we look at that, the black spectrum falls on top of that, or almost all regions of the red spectrum are also covered by the black spectrum. We have some additional regions, and this is not surprising because we have a full C-terminus. So the N-terminus is mainly conserved as far as we can judge that from this mess. And the C-terminus, of course, gives rise to some new peaks. We have mainly threonine, valine, and isoleucine, and also the mainstream amino acids, which give rise to this mess in between here, where we cannot discriminate a lot. So it's not possible to make any site-selective assignments here. But what we can do is we can look at what has been done before by Wüthrich et al. A long time ago, and overlay that with the shifts they get and split that up into different regions. So here we have a lot of different subsets. And I just take exemplarily two of these regions out. Here are the shifts expected for alpha helix 2. And here are the shifts of alpha helix 3 in this region. And what we see is here we see some differences. So the typical alpha helical shifts of Wüthrich et al. Have moved more to beta sheet or random coil shifts here. That means here these two helices are partly affected, maybe dissolved, maybe disordered, maybe not fully fully aggregated or fully destroyed, but that something has happened. So they were affected by this complexation here. That's for sure. And so this is also in agreement. What Harris from Boston observed that these proteins, the C-terminus is really needed for capping and sequestering the A-beta. So maybe it's also interacting closely, tightly with the A-beta oligomers here. So this is what we could see for the prion protein. And if we look at the A-beta oligomers, we can also compare them again to the liquid state spectrum of monomeric A-beta. And what we see here is, of course, these two spectra, they don't match too well. The A-beta completely changes its structure, which is also not so surprising because it's pre-incubated and it is oligomeric beforehand. So we have oligomers, and oligomers are supposedly beta sheet-like, which has been shown by many other groups before, which, who have done also some studies on oligomeric A-beta. And so here for alanine, we see that we are mainly here have moved towards the diagonal, which means more beta sheet-like structure. And here also serine is also away from the diagonal, which is also indicative of beta sheet-like structure. And if we analyze that a little bit more carefully, so first of all, my PhD student, Anna, could even get some site-selective assignments for some parts of the oligomers, but of course it's messy and huge. We get many more signals than we expect, which is also not surprising, because in such an oligomeric preparation where some I-betas are in contact with PRP and some are not, it's not surprising that we have disorder and not all monomers can be equivalent to each other. If we overlay our spectrum with chemical shifts predicted from other structures, then we see the following. We have here different, for example, in red, the mature fibril, about which I will talk a bit later. And then green from Marielle Welti and from the group of Beat Meyer. And then blue from Colvin of Bob Griffin's group. And in yellow, we have also some artificial A-beta protofibril or oligomer from Lendl et al. For live herd. And almost all of these secondary structure also superimpose quite well with this structure. So that means that probably we have a lot of preformed different microstructures which all are present in this oligomer, in this early oligomeric state. And maybe this also might have the potential to evolve into different of the fibril types, which are all there. Okay, we have a different mixture of different beta-strand-rich assemblies, which have some potential to evolve maybe in different fibril types. Yes, and this is here. So what we found out is about these, a little bit about these structures that we have here, some preformed beta-sheet-rich oligomeric assemblies in these oligomeric parts. And in between, we have some semi-rigid prion protein, which is keeps all the oligomers together in this precipitate. And we have some alpha helical ends in the region 130 to 133 or 130 here. Some conformational changes are taking place. Okay, that's about the oligomers. And then to the fibrils. We have looked at A-beta 1 to 42 fibrils some years ago. And together with Gunnar Schröder in our institute, he got a nice cryo-EM structure. And we also did the solid state NMR investigations on that fibril. And what we saw first, before the structure was out, was we really saw one set of peaks, or almost one set of peaks with some slight inconsistencies here in the serine region and also in the phenylalanine region. But apart from that, it was really homogeneous, one set of peaks. And we could really assign one set of peaks to one residue here. That means it was homogeneous. And we could see one set of peaks for every residue. There were not hidden signals. Everything was there. And almost everybody was beta sheet-like. And the most amazing part was also this long beta sheet here from 11 to 22. And I went through the spectrum with my PhD students 10 times and said, it can't be such a long beta sheet. Is that possible? But then when the structure came out, yeah, it's perfectly matched it. And what we also saw is the second puzzling thing was all the chemical shifts of the COO- groups were mainly indicative of a deprotonated state, although the fibrils were grown at pH 2. And this is also, first of all, a little bit strange. Carboxyl groups should have a pKa of 4, around 4 to 5. And at pH 2, they should be protonated, like this aspartate 23 here. And what we then, when we looked at the full structure, bingo, here's this long beta strand, it's there. Then up to the end terminals, it's really ordered. We have some, and all the deprotonated amino acids are really close to a positive group where they can form salt bridges and stabilize the deprotonated state. So everything makes sense here. And yeah, of course, one drawback of the structure or one flaw is, one question that we get frequently is, yeah, this is a pH 2 structure. So could it also exist under physiological conditions? And that's what we tried next. So we tried to, so this was a nice protocol for reproducing fibrils and to get nice and homogeneous fibrils. But what happens if we change then the pH later? Will these fibrils be stable or not? And we tested a wide range of different pH values under which these fibrils could exist. And we added different amounts of citrate phosphate buffer at different pH values and looked at the full range of fibrils. And what we see here is the comparison between both extremes. So what we see here is in red, the sour, the pH 2, in blue, pH 7. And if we look at the spectra at first, we see the peaks did not shift much. Most of the peaks are where they were. If we look a bit more close, we see here some differences for alanine 42. This is quite a jump here. also in the carboxyl region, something has happened. And there are some peaks get a little bit smaller, especially particularly towards the end terminus. But if we look at the inept spectra, we don't see any inept signals. It's still stable and nothing gets really loose. The fibrils are there and the main core is also preserved. Which changes do occur when we look at this alanine? We see here two peaks. And the interesting thing is we have the protonated state, we have the deprotonated state, and the pH values in between. We don't see a move of the peak, a slow move of the peaks, but mainly the red peak disappears and the blue one appears. And at pH 5, we have two peaks. So it's a slow exchange. That means the protonation and deprotonation is slower than the NMR timescale. That means if there are two different ways and something more global happens to the alanine 42 than just the exchange of a proton. And when we look at the intensities of these peaks, we see also, it happens, the transition is between pH 5 and between pH 4 and pH 6. So up to pH 4, nothing happens. So these fibrils would stay in the same state at pH 4, which is also almost a physiological pH in the lysosomes and so on. Okay, and on the other side of the alanine 42, it also makes a salt bridge, or it's close to the salt bridge between aspartate 1 and lysine 28. And when we see here, lysine 28 also undergoes a change from here to pH 7. And it's also slow on the NMR timescale. We see two sets of peaks. Whereas for this other, for the aspartate 7, which is here in this periphery here, and free accessible, we have a slow transition where the peak just moves with a rising pH. Okay, so what does that mean? So probably this aspartate, this alanine 42, has a tendency to get deprotonated somewhere at pH 4, between pH 4 and pH 7, and then it might take the lysine from a salt bridge here and release the aspartate 1. So this is a hypothesis, and that this alanine 42 then gets more involved into the salt bridge and aspartate 1 is released. And we tested this a little bit also with water-acted experiments, which have been done a long time ago, suggested by Samulski et al. Or Kiss and Kumashiro, and also by different other groups. And so we first destroy the magnetization in the protein, then let it flow back from the water to the protein and see where we are. And depending on the mixing time, we can test how close a group is to the water or how well protected a different group is from the water. And of course, it's not only the distance from the water, but it also makes a difference how fast the exchange between the water protons and the protein is, and how well protected exchangeable groups are. And this is particularly grateful method also for such COO- groups, if they are protected or not, and how fast is the exchange for that. So first we did just build-up experiments here at pH 7 and at pH 4. And what we saw here is the initial build-up is already faster for pH 7 for the whole fibril. This can have two reasons. One is that the fibril might be a little bit destabilized, a bit more loose. The surface, water accessible surface might get larger, but also at pH 7, the exchange between protons is faster. So both can play a role here. And when we look closer at it and take 2D spectra and then look at the alanine 42, it's the two pHs. And then we see that here at pH 7, the alanine 42 is still quite well protected with respect to the other CO groups here in the protein. And that means it really is somewhere buried in a salt bridge and not freely released into the water. And this is also a hint that this alanine 42 still is the main player in the salt bridge, even at high pH values here. And that this salt bridge is rearranging here. Yes, and then we have some colleagues who did some molecular dynamics studies, and what they saw first, they could also confirm that the salt bridge is rearranging, And second, they saw no significant differences in stability between pH 2 and pH 7 when they measured the RMSD between different structures after different times. So nothing, it does not get very unstable here. So it's in agreement with the NMR results that the global fold of the A-beta fibrils is unchanged even at high pHs up to 7.4. Then there are no significant changes, no changes at all up to pH 4, and local changes occur starting from pH 2, and especially for residues in the salt bridges, so some stabilities get affected, and of course maybe the N-terminus is not that fixed to the structure anymore than it is at pH 2. And this is also maybe a reason why we see this stabilized N-terminus, this beta sheet core N-terminus in our pH 2 fibrils, whereas in other structures which were grown at pH 7, it's not there. Okay, so this is mainly not such a big difference here. Okay, so this is the end of my talk. I would like to thank all my co-workers, and yeah, this picture is from better times when we could still stand that close, but yes. So these are all our co-workers and colleagues with whom we collaborated. And thank you for your attention.
Thank you very much, Henrike. Nice talk, and for staying so perfectly in time. Thank you very much for that.
Did I? Yeah.
So I see questions from the Q&As. Maybe we start with that one, and maybe some others can also ask some questions. So what is the reason for doing proton spin diffusion instead of DAR? That's the first question we got.
Yeah, I think this is a matter of taste. At 11 up to 15 kilohertz, you don't need DAR. We tried it many times, and we never saw a difference between DAR and proton-derivative spin diffusion. At higher spinning speeds, it might have an effect, but we never discovered any.
Okay. Then we have a question from David Elisa. Welcome, David. Great to have you. Do you think that losing the intermolecular salt bridge at neutral pH might increase the stability of the protofibril interface and result in fibrils with only one protofibril?
Okay, that is interesting. I think, yeah, if we look at the full fibril, I mean, after having this fibril, it's really tightly intertwined with two different stages. I think it will not rip apart these fibrils. They are wound around each other, and all these hydrophobic interactions here between those two interfaces, I think will still stabilize the fibril strongly enough not to fall apart immediately. And we also did not see any changes over 1 year, over the course of 1 year in our spectra. So I don't believe that it will fully fall apart. And this salt bridge here, yeah, it can be between one. I don't believe that it will be enough to fully destabilize everything. But of course, at pH 7, maybe probably the other arrangement is more stable, as we saw, as we get a different type of fibril then.
Okay, thank you. And another question from Entos. Do you see pi-pi interactions between the side chains, I assume?
Pi-pi interactions between the phenylalanines, probably.
I guess so, yeah.
Yes. What we saw is, yes, this, we had some, I didn't talk about that. I said our spectra are quite nice and homogeneous, but there was 2 different sets of peaks. We saw some alanine, one phenylalanine peak, 21 and 20, I think, 21 alanine. And it's not in the spectrum visible, but we saw some different conformations there. And actually this phenylalanine 20 can have 2 different conformations. And we also saw in the cryo-EM, there were 2 electron clouds here and here with equal population. So the phenylalanine can adopt 2 different orientations, and that affects the alanine 21 shifts quite significantly. So we saw 2 sets of peaks here, and I guess this is also of the pi cloud, a shift from the pi. And I guess pi interaction, probably that also stabilizes the intermolecular contact between the fibrils, I guess, when they are on top of each other.
Okay, then there's a question from Rob Tico. Great to have you, Rob. Does the pH, that was actually also one question that I had. Does the pH2 fibril structure ever grow spontaneously? It has de novo at pH 7. Can you use the pH2 fibrils as seeds to grow fibrils at pH 7? Is the structure preserved in the seeded growth?
We haven't seeded yet. That would be an interesting experiment. De novo, I'm not sure. So in some of the oligomer samples, when we let it sit for a very long time, so we have here this valent 24, these 2 very pronounced different shifts of C alpha and C gamma, which is here. And this has never been observed in the other structures. And we saw that when we leave our A beta PRP samples for a very long time, those 2 peaks appear also in those, and they are not at pH 2. So I wouldn't exclude that it is at least some parts or some conformations of this area here can be adopted also at higher pH values. But of course, it's not the full fibril here.
Okay. Now a question that also came to my mind, the temperature shown in all your experiments. Is that the probe temperature or the sample temperature? And actually my question was especially for the oligomeric samples that you measure. I mean, are the samples frozen then, or could you still see some disappearance of peaks because it's just too mobile for the...
And we could still see, it could be that some peaks are still mobile. So it's above the water freezing temperature for sure. And I think I gave the temperature that we aimed for. No, they're here in this. Yeah, I think we try to correct for the. So
in the oligomeric state, you had 0 degrees and 11 kilohertz MS. So that means they would be frozen.
We were 0 degrees sample temperature. No, water. It's not pure water. It's very salt rich and protein rich. So it's not frozen yet. It doesn't freeze up to a sample temperature, nominal temperature of -30 or so. So the water peak is still small and narrow.
But could that not mean that you then, because it's a dipolar-based experiment, I guess, right?
Yes.
When you're looking at that, some of the peaks that are missing are just missing because the protein is still mobile in that region?
That could be, yes. We cannot say that we really see 100%. We do not see much more. We do not see any distinct peaks in the direct excitation that we don't see in the dipolar. But it could, of course, be that some of the regions are also not visible. So this is also, if some helical peaks do not appear here, we cannot exclude 100% that it's not because of some residual mobility here.
Mm-hmm. And there was another question in the prion oligomer experiment. Why is the carbonyl signal temperature dependent in the CP spectrum, but not in the direct excitation spectrum?
Temperature dependent. Okay. Yeah, maybe. So I think these experiments were not optimized for CP. I think it was with a short CP, and maybe the different mobility, residual mobility puts it, if we cool down to -10, maybe the colder temperature adds a little bit to the rigidity to make it a bit more sensitive to the CP at lower temperature. So I think maybe we could also get better CP spectra or higher carbonyl value signal at 30 degrees if we would optimize a little bit more for that. But of course, yeah. So the mobility increases, of course, with temperature. And I guess we see that because the dipolar couplings are just weaker here. That's my guess.
Okay, I see another question about the pi direction, but maybe that's something for later that you can type the answer to the colleague. NTOS. I don't see any other questions. Are somebody from the panel? May Missy? You look like you wanted to ask the question to me. May Hong? Oh, I can't hear you.
No question for now. The informal session.
Okay. Okay, if there are no more questions, I think I give the word back to the organizer, Marcus. You want to? Thank you again for the talk.
Thanks to everybody for excellent talks and also for the introducers and moderators. And then we officially close it for today. And